Hello and welcome everyone. We're giving everyone a few seconds to join the meeting from the waiting room. I see participants are joining, and as always, I see familiar names as well as new people. So welcome to everyone who has been following us on a weekly basis for a while, and also welcome to all new people. This is our weekly webinar update about the HEALEY ALS Platform Trial. And tonight I'm particularly excited to have as a guest speaker one of our wonderful site investigators, Dr. David Walk from the University of Minnesota. We've been working together on several projects for many years, and I'm so excited that you are the site investigator for the platform trial, your site. If we can go to the next slide. Again, I'll just turn it over to David to tell us more about his experience in the trial. David?
Great. Well, thank you so much. It's a pleasure to be able to participate. And apologies for having my face covered, but I'm working in an office in the clinic building. Of course, I'm required to remain masked. So you have a slide that shows you what my lower face looks like. Why don't we move on to the next slide, and I'll introduce you all to our team. We have 2 to 3, if you like, coordinators for the platform trial. I always like to have a backup coordinator for a study, but in this study in particular, with the level of complexity and the level of interest, it's been wonderful to be able to have a team that's all working together. Valerie Vermont has been working with us in ALS research for many years now and is an extraordinarily capable coordinator and doesn't miss a thing. So she's really, really good at keeping me on my toes and making sure that people are participating in the safest and most compliant way possible. Kat Kokel worked for us for several years in the past and went off to cardiology, and I'm happy to say has recently come back to neurology. And the two of them have very complementary styles and make for a great team. Carly Schrader is a research coordinator, but principally working as an evaluator. So as everybody knows in this trial, it's particularly important to have somebody who can focus well on the pulmonary function studies and some of the other outcome measures. Carly has a background as a psychometrist, somebody who has been doing cognitive testing for several years and is very precise and careful and does a wonderful job in that role. And again, the 4 of us, just when we've got participants in the study, we just get together and basically stay for the morning so that we're all sort of available to help each other out. The other extremely important member of the kind of on the ground team is Dr. Miser. Sam Miser trained in neurology and in neurophysiology in our department here, and then did a year of fellowship in palliative care and runs the ALS multidisciplinary clinic at Hennepin County Medical Center, which is one of our sister clinics. And he's just an extraordinary physician. And of course, his training provides him with all of the tools to be an outstanding ALS clinician. So, and just a first-class human being. So he's been a wonderful partner to have here. And we'll talk a little bit more about the partnership in a moment. And finally, Sarah Hilbert's been working in our neurology research area for many, many years, started out as a coordinator and is the person who does all the behind the scenes work and makes sure that all of the kind of regulatory and financial things get done and get done correctly, and is a tremendous asset to the team. We'll go to the next slide and I'll kind of introduce the concept of how we collaborate. So you'll notice that these people are not at work. This photo was taken at this fall's ALS walk and represents staff from 3 multidisciplinary clinics in the Twin Cities. Ours at University of Minnesota, Dr. Miser's group at Hennepin County Medical Center, and Dr. Gugliani's group at Health Partners, which is a large healthcare organization here in the Twin Cities, and their clinic is in St. Paul. All 3 of us have excellent multidisciplinary ALS clinical programs, but we recognized years ago the importance of collaborating on research. Different institutions can be, as businesses, they can be competitive on some level, but the providers in those institutions need to be working collaboratively on something this important. So We meet monthly, speak regularly, and actually at the ALS Association have a quarterly conference here in the Twin Cities. The physicians at all three are faculty members at the University of Minnesota, which makes it a little easier to collaborate. But we work very hard on making sure that everybody who is a candidate for and interested in clinical research has an opportunity to participate. And if they're not a candidate at one of our sites studies, then we inform them about the other studies and make sure that people have all the opportunities possible. And similarly, by having Dr. Miser enroll patients or participants in the platform trial at the university, and conversely by myself being a co-investigator in his studies, we're able to follow our own patients, so that if he tells somebody at his clinic about the platform trial at the university, he can be the physician who evaluates them and enters them in the study, which I think provides some confidence, some comfort, and some continuity for individuals. Dr. Gugliani will hopefully be joining soon as well as an investigator in the platform trial, but we're very proud of the fact that we work collaboratively across our metro, so that we're all kind of focused on the same ultimate goal, which is finding better treatments for ALS. Again, I appreciate the opportunity to participate, and I'll join in the rest of this, but I must say it's been just a great pleasure to be part of the platform trial, and it's just it's an idea whose time had long since come, and we're also grateful, of course, to Sean Healey and to MGH for getting this going, because this is going to accelerate our ability to find better treatments for ALS. And you've already made such great progress in 3 arms in such a short period of time. That's not the sort of thing that normally happens in clinical research. Thanks again, and I'll pass it back over to Dr. Paganoni.
Thank you so much. So for everyone who's in the Twin Cities, who should they contact? Would that be Valerie or?
Either Valerie or Kat. I believe
Perfect.
they handed out the same number. I had not provided their number just as an oversight, but perhaps that can be done by email.
Yeah, and Catherine, if you don't mind putting all of that in the chat. So I really love the fact that you and Dr. Miser collaborate so closely at the Twin City area. So that's fantastic. And so in terms of, it's really because of the work of sites like yours that we've been able to make progress in this trial. At this time, as you know, we are enrolling participants, testing participants to see the safety and efficacy of 4 drugs that are listed on the slide. And now we have already several participants who have completed testing in the randomized placebo-controlled trial and have continued on open label extension. So if we can go to the next slide, we can just share our weekly update. It's really amazing to see that over 800 people with ALS have signed informed consent. So I just really wanted to take a moment to think about this number and really thank everyone who's participating or supporting friends or family as they participate in the trial, or just even raising awareness about the trial. All of this counts and really helps ALS research to move forward. We had patients follow, enter the protocol, and then they're assigned to any of the regimens or drugs that are being tested. At this point, over 500 people have already entered the trial in terms of being assigned to either active or placebo, and over 200 people have already completed the placebo-controlled trial and entered the open label extension. You can see the breakdown in terms of the regimen assignments there, and as for those of you who follow us on a weekly basis, you know that our target enrollment for all regimens, for each regimen, is about 160 people. And you can see that we are basically essentially at target for regimens B and C, and we're rapidly approaching that target for regimen A as well. We started regimens A, B, and C at the same time last summer. And so essentially this first group will have results at around the same time next year, and then regimen D started 6 months later, but definitely is making a lot of progress. And as A, B, and C fill up, essentially D will enroll rapidly after that. So we're really excited that we're going to have essentially the results of 4 drugs, sort of closing time. Again, the first 3 together, and then D to follow shortly after that. So that's really amazing to have results of multiple for multiple drugs, not just as one, which is really a change in the field. If we can go to the next slide. This is information we share every week. We have a lot of information on our website. We even have QR codes. We post all of this information on our website as well as the slides. So feel free to go back to our weekly recordings or slides from previous webinars. You can always go back and check the sites that are participating. At this time, we have 52 sites that are actively enrolling, and we are in the process of adding over 20 more sites. So we will continue to update the list as new sites are added. Next slide. For any question that you may have, we have Catherine with us today. We also have Alison. They're our patient navigators. You can always contact them by phone or email with any questions. Next slide. And we continue to have weekly webinars so that we continue to update the community about our progress. Next week, we will have one of our investigators from Penn State. So we're going to move to a different state next week. It's really been wonderful to get a flavor for different clinics and really share names and faces of people that are the boots on the ground and really enrolling for this trial, people that you can contact depending on your geographic location. And we are also very excited to have Dr. Miller from NIALS join us on October 28th. As some of you know, as we announced in previous webinars, we also launched an expanded access program companion to the platform trial that's funded in part by NIALS. So we're very excited to also have her join one of these webinars as a guest speaker so that we can talk more about that initiative as well. Next slide. Oh, that was the last slide. Great. So at this point, I think we can take questions. So feel free to type questions in the Q&A box, and we'll take them as we get them. Also had a few questions that are coming in. I know we've been receiving questions about other studies as well. And maybe Dr. Cudkowicz, I'll ask you this first question about a separate research study. The question is, a Kyoto University-led research team said that its studies on stem cells discovered that there's a new medicine called bosutinib that halted the progression of ALS in patients for the first time. So just wanted to, I know this was recently in the media. Can you comment on that research?
Sure. I think the preclinical science is really good. They can take blood or skin from people with ALS, and they can make their motor neurons in a dish and then look at drugs that might improve how those cells do in the dish as a way to screen drugs. And they came up with this drug, bosutinib, which is on the market for a type of cancer. And then they did a very small open label study, open label meaning no placebo group, treated a few people with it. There's some people couldn't tolerate it and stopped the drug early. And then the few who could take it without any side effects, they reported that they did very well, what they called stability. First of all, anytime there's any news about someone doing well, I think it's always positive news. You don't know where the findings are going to come from. And this is good science, but you have to also be very cautious about these small open label studies, because we've been here before with reports like this, that then when you actually do the trial, don't pan out. So I hope that's not going to be the case here, but I think the next step, and I know they're planning this, is to do a trial of that drug and bring that forward and see what it does in ALS.
Great, thank you. There's a few questions coming in. Are there efforts to perform genetic analysis of patients to look for mutations beyond the two common ones? So not just SOD1 or C9, but I guess I think the question is in general, I guess both in the trial and in the clinic. So maybe I'll ask both of you to comment on this.
Sure, David, Dr. Walker, if you want to take that first. You're on mute though.
So was the question regarding clinical trials that are directed at other mutations, or simply the question about testing people living with ALS?
I think the question was about testing. Yeah.
Right. No, that's a huge question, and we are moving, I think, more and more towards testing more generally for a couple of reasons. First of all, we are learning more and more about the genetics of ALS with time, and there are different views of it. There are those who say, well, gee, if you don't have a particular treatment for that type of gene, why would I want to know? But number one, there are certain circumstances that are unusual and that make us wonder about genetic form. So for example, somebody with an unusually early presentation, early in life, young presentation, makes us think about certain genetic forms. Or somebody whose family history is not that complete. Maybe they don't know much about one parent and their family, or circumstances in which there might be some features that we associate with certain types, genetic forms of ALS. For example, association with dementia or psychiatric disease in the family, or a pure lower motor neuron form that might be moving rather quickly might fit with certain genetic forms. Now, why would it matter? Well, as you know, there are clinical trials that are specifically directed at particular genetic forms of ALS, and at least one that is well along, shall we say, we'll learn soon about efficacy. But the moment we know that there is a specific genetic form of ALS that has a unique treatment for that form, it will become imperative, I think, for people to be tested generally, even if it's something that's rare, a 1 or 2% chance. Then we're going to want to identify everybody with a mutation in that particular gene. And I should add, there are also expanded access opportunities. For example, FUS mutation, which is extremely rare because it's a very serious, particularly serious form of ALS, is one in which there are expanded access or clinical trial opportunities. We don't want to miss those individuals. So I think that we are moving more and more towards testing many, most, or perhaps everybody living with ALS, so we know as much about them as possible and have the opportunity to enroll them either in gene-specific clinical trials or get the medications once they're approved.
Great. And just because of your comment about expanded access, there's a nice comment in the chat about sort of thanking Dr. Cudkowicz for your testimony at the congressional hearing about different pieces of legislation, including expanded access and the Act for ALS. So the comment is about the fact that we're close, you know, it seems like the Act for ALS might be passing in the House because there's a lot of sponsors, and this would represent more funding, you know, for expanded access programs. So big thanks to you for your advocacy, and any comments you may want to add about EAPs.
I just want to add, first of all, it's a whole community that has gotten us to the point where we're so close to getting that bill passed, as well as the other bills. So that's fantastic. I have to admit that I never thought I'd be talking to my 2 senators' office to try to convince them about anything. Didn't know if I could threaten not to vote for them. I'm not sure it makes much of a difference in my state. But anyway, they saw that their staff saw the light, and I'm really glad that they're supportive.
Great. There's a good question about geography and coverage. So the question is, there's about 20 states without a site just by looking at the map. And so the question is, when we add over 20 sites, does that mean that we will close this gap, or how is that going to work in terms of state coverage?
Yeah, that's a great question. Yes, we're absolutely going to close the gap. I have to confess that I don't know exactly how many of the missing sites we're filling, but that was one of our goals, was to pick in areas where there wasn't enough coverage. We did add, you know, though, also some sites in states where there was only one, but huge populations, such as New York. But other than that, we were really trying to get into new areas. We'll show very soon, maybe at one of these upcoming meetings, where those additional sites are located.
Great. And there's a question for clarification. Catherine, can I ask you to put in the chat the name of the cancer drug that was used in the Japanese study, because one of the participants wanted to get the right name to look it up. Thank you. So there's a question about the upcoming regimen. Actually, thank you for pointing that out. I didn't mention during the presentation that in addition to completing enrollment for the first 4 regimens, we will soon be adding regimen number 5 from SILOS. And the question is, Dr. Cudkowicz, will we hear more about this new drug on upcoming webinars?
Yes, we are planning to have a 1-hour webinar where we have the company come and speak for maybe 15-20 minutes and answer your questions, like we've done for A through D. Whenever we have a presentation about the actual trial, we need the slides reviewed by our Central Ethics Committee. So that's happening. And as soon as those are approved, we'll set the date and let everybody know.
Great. There's a comment about Neuron, the stem cell investigational product by Brainstorm. The comment is that it's been tested also in progressive MS or progressive multiple sclerosis. And so there was a comment about, I think a presentation that was given at the MS conference, and the fact that there seem to be some positive results. Any comments, Dr. Cudkowicz, about the correlation between ALS and progressive MS? Is there anything in common between the two diseases?
I think that they are very different diseases clinically and biologically, but what they do share in common is an inflammatory reaction to the illness. In progressive MS, it might be more, that inflammation might be actually, more upstream, but both illnesses have this inflammation, and that's what neuron, the mechanism of action of neuron is supposed to be anti-inflammatory. So that's, I think, why they were trying it in both illnesses.
Great. I wanted to ask you a question, Dr. Walk. There's a great question from a potential participant in the trial. So the question is, where would a potential participant find information about the potential risks of the drugs that are being tested in the trial?
So there's probably, and again, you and Dr. Cudkowicz can speak to this, but relatively little public information because these drugs have relatively little track record. And actually, as an aside, I think that's one of the exciting things about this trial. We're looking at drugs here that have very little exposure to people living with ALS, and therefore potentially, great opportunity for new observations and new discoveries. Now, part of the process of participation, of course, part of the consent process is to review potential adverse effects, and that information is detailed very extensively in the information that we as investigators are provided. So I can sit down and speak in as much granular detail as one would like about how many studies have been done, how many people with what conditions have been exposed to these drugs. The consent forms, of course, talk about the potential for various adverse effects, but it's important to keep in mind that we also have information on the number of people who were on the placebo arm of those studies who had similar potential side effects. So the important thing to keep in mind is that these are relatively new agents. They have been studied extensively in animals and in human volunteers. So they wouldn't have gotten to this point if there weren't a reasonable expectation that they were fairly safe molecules, but we're always learning more things. And so I think the most important thing is to have as much of an in-depth conversation as you'd like with the investigator at your site.
Right. Question about one of the regimens, regimen C, is testing one drug produced by Clean known as CNM-Au8. And as some of you know, there's a separate small study in Australia called Rescue ALS testing the same drug. This is much smaller than the platform trial. Dr. Cudkowicz, any info in terms of when the results of that study will be published?
Yeah, thank you for asking. I think before the end of the year. We haven't heard specific dates on that, but I'm hoping that it's going to be soon. Now it is a small study. I think it's 50-50 drug or placebo, about 25 people, I think, in each group, or a little less than that. And they're looking at electrophysiology measures, so measures of how your motor nerve connects to your muscle, and how that connection. So I think, of course, every information is helpful, but I'm not sure that it's going to change like what we're doing. We've been talking about this a lot, given the size of it, but obviously any news is good news. So we are waiting for those results.
Thank you. There's an interesting question. It says, if hypothetically, if a drug in an early phase 1 clinical trial had clear halting or reversal of disease, is there precedent for making it available without additional testing in phase 2 or 3? Do you know if that's?
Yeah, I think that, well, it depends if it's a well-done study, meaning that it's controlled and you can interpret the results. I think there is. I mean, I guess the biggest example, I don't know if it was a phase 1, but the first drug for HIV, I think the first time they looked at the data, safety monitoring board looked at the data unblinded, there was a huge imbalance with, I think, 10 people passed away on the placebo group and none on the treated, and they halted that trial and approved the drug. So if it's a dramatic, like what you're describing, and it's a well-done controlled study and they see such a big difference early on, that they could go right to approval.
Great. There's a question about any updates on the AMX35 trial. So I just wanted to mention that we have the recording of a webinar that we gave just 2 or 3 weeks ago. So I'll ask Catherine to post that. So briefly, basically, the company Amelix Therapeutics has announced that there's going to be a submission to the FDA based on the results of the CENTAUR trial of AMX35. Obviously, that's great news, and we're all excited. So again, for additional information, I think we gave a good description in the webinar, but feel free to come back on this webinar and ask follow-up questions if that was not clear in the previous webinar. There's another question again about CNM-Au8, again, regimen C tested in the trial. So this drug is also tested in multiple sclerosis in a separate trial. And there was some recent announcements about blinded interim data. So essentially the company released some information as the trial is still ongoing. So we don't have the final results. Dr. Cudkowicz, any comments about the testing of CNM-Au8 in multiple sclerosis?
Yeah, I think we just don't know for 2 reasons. One is that it is a different disease, and let's say it doesn't work in MS eventually. I don't think that means it won't work in ALS or the flip, because they really are 2 different diseases. And I do think that looking at blinded data is maybe helpful for safety, but you can't really say much about efficacy. I know that Clean has done this now for their rescue study in the MS, but until you really know who's on drug and who's not on drug, it's hard to interpret anything about efficacy.
Right. So there's a question about sort of mitochondrial dysfunction and more the molecular mechanism. So essentially the question is whether the demyelination can occur to free iron from the myelin to boost production of ATP. I know it's sort of a technical question, and the question is whether, I believe the question is whether, mitochondrial dysfunction might then be involved, and that might be a cause of ALS. Any comments on that, Dr. Cudkowicz?
Yeah, I was hoping you were going to give that one to Dr. Walk. I know when to admit my ignorance. I actually don't know the answer to that question. I can ask one of our MS people or David if you want to weigh in.
Yeah, I mean, so I think that you do know this, but just to be a little more explicit about the nature of the question. So, the role of myelin in ALS, we don't know very much. We don't think of it as kind of a major player in the pathogenesis, but we do know that mitochondrial dysfunction is part of the problem. And I would just say, I hope you both agree that when we talk about ALS, there's some excellent summary diagrams from review articles that show what goes wrong. And they will show mitochondrial dysfunction as well as abnormalities in just about every other part of a motor neuron. And it's, I always say that when the diagram is that complicated, it means we don't know enough about the disease yet, because we still don't, of course, understand what starts the process, what's cause and what's consequence. But certainly mitochondrial dysfunction is part of the problem. One of the reasons we're still here is that we need our basic scientist colleagues to really give us a better overall understanding of what are we dealing with here.
Great. So I do want to take one last question, which I think is very timely. Last week, we didn't have this webinar because we all participated in the annual NIALS Conference, which is the annual scientific meeting of the Clinical Trial Consortium, where we enroll participants for the platform trial. So I guess for both of you, I wanted to, a great question came in, what are the, any news, any relevant news you would like to share from the annual NIALS meeting that happened last week? Anything that's cause for optimism? What are your main takeaways? Maybe I'll ask Dr. Cudkowicz first, and then Dr. Walk.
First of all, it was a fabulous meeting. I mean, it would have been better if we were all together on the beach in Florida, but it was fabulous. And there were a lot of people and individuals with ALS and family members there, scientists. It was great. 2 takeaways. One is Dr. Rothstein at Hopkins presented some amazing work trying to understand why the nuclear pore, which is how things get in and out of the nucleus in your cell, why it's not working well and why it's leaky in ALS. And if you guys remember, regimen D is a drug that works on the nuclear pore. So he's found a new protein that's not working well, and he's already working with Ionis on a gene therapy to fix that, and that would be for sporadic disease. So that was just amazing science. And the other takeaway is that EAP is now the new lingo. Like there was a lot of discussion about that. A lot of centers that want to open up programs on this, and they've heard loudly that this is really important for the people that they care for, and I thought that was a big change in approach. So I was excited about that.
Great. And Dr. Walk, I'll leave you the last word to conclude this webinar. Any insights from the NIALS meeting?
Well, my last word is that when it comes to summarizing a NIALS meeting, I'm never going to step on Dr. Cudkowicz. I'll let her talk about that. But no, you're clearly helping us all make progress, and we'll get there. It might be incremental, but we'll get there. It's a terrible disease, and I can see that I think we're going to have more breakthroughs faster now than we've ever had, and it's a very exciting time.
Great, thank you. Thank you for that, and we will see all of you next week. Thank you.
Bye.
Thank you. Bye-bye.